Hello everyone. Thank you for tuning into this presentation. My name is Dr. Fiordilici Gatilog, Head of Marketing and Scientific Communications at PEPperPRINT, and today I'd like to tell you more about our company and our high-density peptide microarrays for antibody validation and vaccine development. Just to briefly introduce our company for those of you who might not know us, we are founded and based in Heidelberg, Germany, and had our first product launched commercially in 2011. We are a privately held independent company made up of an experienced team of executives. We provide our products and services to an international clientele, working together with both the industrial and academic sectors, for which we have an excellent track record. We are also ISO certified for quality management. Currently, we have about 25 employees, in addition to an expansive sales and distributor network across 5 continents. At the core of our business is our unique laser-printed peptide microarray technology. To put it simply, it's like what you see here, we can print out practically any protein as highly overlapping peptide libraries onto glass slides, which may then be screened against sera or antibodies, and later analyzed to identify peptide sequences of interest. All of this is made possible thanks to our award-winning platform technology. We have a laser printer that allows for direct printing of amino acid particles onto glass slides. This printer consists of 24 printing units, each corresponding to the different amino acids. Through cycles of melting and coupling reactions, amino acids are printed one layer at a time, building up peptide chains, and eventually full peptide libraries as seen in the diagram here. Now, what's great about this method is that it minimizes both material consumption and production times, which directly translates into decreased cost and maximum flexibility without compromising quality. Our platform also allows us to print both linear and cyclic constrained peptide libraries, which allow for screening of both linear and conformational epitopes respectively. And because we have that digital printing flexibility, we can produce our PEPperChip peptide microarrays in a wide range of formats, which can both accommodate a variety of experimental designs, as well as be used as an investigative tool for a range of research applications. Our peptide microarrays can be readily adapted to support your research aims, from large antigen discovery screenings, as with infectious or autoimmune disease research, to more targeted epitope characterizations, as with antibody validation. We also offer a range of solutions for our customers. In addition to producing standalone PEPperChip peptide microarray products, we also provide full fee-for-service packages, which includes everything from immunoassay to data analysis and reporting. We also have our own R&D projects with a focus on biomarker discovery for disease-specific epitopes, which we offer for either out-licensing or co-development. For antibody analysis and characterization, we offer linear and conformational epitope mapping services. We map hundreds of antibodies each year, working with 9 out of the 10 top pharma companies. Now, just to cite a few examples of our epitope mapping case studies, here's one from 2017 we did for MSD, where we mapped epitopes of anti-CMV antibodies. And here's another we did for Janssen Diagnostics, this time where we performed proteome-wide epitope mappings to identify immunodominant peptides in a parasite. We've also worked in cooperation with antibody-producing companies. For example, what you see here is an application note jointly published with Progin from Germany for a project where we validated their anti-keratin antibodies through epitope mapping with our peptide microarrays. Now, as mentioned earlier, in addition to providing services, we're also working on developing epitope biomarker libraries, in particular for autoimmune and infectious diseases. We have achieved this for Zika virus and have successfully filed a patent for our Zika virus-specific epitope biomarkers at the end of 2018. Now, just to share a bit about these Zika biomarker epitopes, these are derived from the Zika NS2B protein, which we found to be among the common core epitopes in all the tested Zika virus patients. And these had no cross-reactions with other flavivirus infections. These biomarker epitopes include peptides of linear, cyclic, wild-type, and optimized variants, as well as false positive control peptides against other flavivirus infections. We've already initiated partnering with the diagnostic industry, and we would be happy to discuss out-licensing and IVD development possibilities with interested parties. So please don't hesitate to get in touch with us either through the partnering platform or via the contact information that we'll show later at the end of this presentation. Now, another really useful thing we could do with our peptide microarray printing platform is to be able to quickly design and generate pathogen antigen libraries for epitope-level antibody response monitoring. Our peptide microarrays can be used to screen for neutralizing antibodies, find immunogenic epitopes, or to evaluate the performance of vaccine constructs and adjuvants in terms of eliciting specific and lasting antibody responses. Epitope information can also be leveraged for a more rational approach to vaccine design. And because peptide microarrays can provide an in-depth analysis of antibody responses, it can very easily be used to study different vaccine types against different diseases. Now, let me share with you a few vaccine research case studies. Here's a phase 1 malaria vaccine trial that was published in Nature earlier this year from researchers from Heidelberg Hospital and Sumaya Biotech. So in this study, they looked into the efficacy of a vaccine candidate which uses a full-length malaria surface protein in eliciting antibody responses. Peptide microarrays containing the surface protein as overlapping peptides were then used to compare levels of antibody responses from different patient and sample groups. And among the study's major findings is a strong correlation between epitope-specific antibody responses and the ability to inhibit parasite growth. Now, what we know is that phase 1 has already been completed, and they're currently preparing for submission to phase 2 trials. Now, here's another example of the use of our peptide microarrays in vaccine research, this time in screening for neutralizing epitopes against CMV done by the researchers at the Icahn School of Medicine. Here they found that broadly neutralizing monoclonal antibodies can recognize a highly conserved epitope on the CMV envelope glycoprotein that they suggested to be potentially good vaccine targets. And here are a couple more examples of post-vaccination immune monitoring studies against different viral pathogens. Here with chikungunya virus infected and vaccinated individuals performed by researchers at the Paul Ehrlich Institute, and with Ebola virus infected and vaccinated individuals in a more recent study from researchers at the Bernhard Nocht Institute for Tropical Medicine. Now, of course, no discussion on vaccine development research nowadays would be complete without mentioning SARS-CoV-2. With the scientific community stepping up and releasing the first viral genomic sequences early this year, we were able to respond just as quickly thanks to our unique platform. We were then able to design, produce, and immediately launch our SARS-CoV-2 peptide microarray within a month of the sequence publication. So we were the first to really be able to provide the whole proteome microarray of the new coronavirus. And this microarray could then be used to analyze antibody responses in patient sera. Now, as a follow-up response, we also later launched the pan-corona spike protein peptide microarray, which includes all human coronavirus spike proteins, including that of SARS-CoV-2, in one microarray chip, which can then be used to analyze antibody cross-reactivity. More information about both microarrays can be found by visiting the links shown here. Now, as we were among the early providers of COVID-19 serological analysis tools for research, we took the opportunity to reach out and team up with Professor Christian Drosten's lab at the Institute of Virology at the Charité in Berlin, who had access to the first patient sera earlier this year. This led to a joint study focusing on IgG and IgA antiviral responses in COVID-19. Now, just to share a bit of that initial data, here you see some of the representative microarray scans. PCR negative control sera on the left, and PCR positive for COVID-19 sera on the right. Samples were screened with a SARS-CoV-2 proteome microarray, which contains a total of 4,883 peptides spanning the length of the whole proteome of the virus. Each peptide is 15 amino acids long, with a 13 amino acid overlap, allowing for high-resolution mapping of anti-SARS-CoV-2 antibodies. What you see here is a dual isotype mapping with IgG antibody responses in red and IgA antibody responses in green. And the chip is framed by HA and polio control peptides. And here you see the corresponding heat maps, where you can also clearly see differences in antibody response patterns between control and patient sera. Now, here you can see a direct comparison of individual samples. We've shifted the baselines a bit here to give you a better overview of the data, but what you can see in this graph is basically IgA and IgG antibody epitope identification on the amino acid level, which you can see is also very different for healthy controls and COVID-19 sera. Now, at present, we've actually completed analysis of additional samples from different patient groups, and we expect this data to be published soon. We're also looking into sera of SARS-CoV-2 recovered patients, and also looking into comparing antibody responses against linear versus cyclic constrained peptides. Meanwhile, multiple biomarker and vaccine studies using both the SARS-CoV-2 and pan-corona spike protein microarrays in the US and Europe are currently underway. Now, for those of you who may be interested in learning more about how our SARS-CoV-2 proteome microarray is being used in COVID-19 research, please do take a look at these first user preprints that came out in the last few months. Here's one from the Max Planck Institute of Colloids and Interfaces in Potsdam, which talks about the relevance of IgA responses in determining COVID-19 severity. And here's another from the UNC School of Medicine, which looks into different SARS-CoV-2 epitopes for vaccine development. So, just as a take-home message, to summarize what we at PEPperPRINT can do to support your research and development efforts, we can provide you with any high-density peptide microarray in the shortest possible time, and this can be done for any emerging pathogen or disease. Various service options are available on request, and we can accommodate both small pilot studies and larger scale studies. And of course, we provide high-quality microarrays and services under ISO certified quality control. And with that, I would like to end my presentation. For any questions or inquiries, please don't hesitate to get in touch with us either through the partnering platform or by sending us a message at info@pepperprint.com. You can also visit our website at pepperprint.com for more information or connect with us on social media for updates. Thank you very much for your attention.